Hi, everyone. My name is Jesse Isaacman-Beck, and I'm a 2017 Beckman Postdoctoral Fellow. I work in Tom Clendenon's lab at Stanford on molecular neuroscience, and I'm in awe of the brain, especially with how behavior and consciousness emerge from biology. I'm amazed by the many different types of neurons in our brain. They all have the same genome, and yet they tailor gene expression in order to give specific function. I want to define how different types of neurons use the same genes. And what are the differences, and are there common themes across cell types? In my application to the Beckman Foundation, I propose to build genetic toolkits that would allow me to answer these questions. And today I'll tell you about SPARC, a toolkit that I built to express or delete genes in fractions of any cell population in order to define and compare how individual neurons work. Now, I'm particularly excited to give this talk to the Beckman family, as I'm inspired by Dr. Beckman as an inventor. As many of you will remember, we celebrated the 40th anniversary of the Beckman Foundation last year with the motto, "When life gives you lemons, make a pH meter." And we honored how Dr. Beckman invented the pH meter to help a friend standardize the acidity of lemon juice. That's amazing. And as Dr. Beckman said, many others say I've done far more for science in general by making instruments available for thousands to use than just by what I could have done in my own laboratory. Now, I've thought a lot about this with my own work, and while I want to be clear that the tools that I've generated are not on par with the pH meter, I hope that they aid scientific progress well beyond my own interests and inquiries. And I'm deeply grateful to the Beckman Foundation for taking a chance on me in this project. So my lemons are neurons, and I want to define how different types of these cells process information. To do this, we need to observe single neuron function. And when I started my postdoc, we could express or delete genes in whole cell populations, but it was very challenging to observe and manipulate single neurons within these populations in high throughput. For example, here's a neuron type that I care a lot about, and it's expressing GFP. You can see the cell bodies in the top and the neurites and synaptic arbors below. There are about 750 of these MI1 neurons, and they exist in repeated circuits in the fruit fly brain to process information, visual information. These MI1 cells are some of the most separated cells in any brain, but they interdigitate, and when we label them all, it's really hard to observe single neurons and single neuron function. I wanted a way to reproducibly target non-overlapping neurons in this population and any population. And I also wanted a way to delete genes in these neurons after development to determine how the adult neuron uses these genes. And again, to be clear, I wanted to be able to do this in any cell type in order to compare across neurons. Now, in my mind, the fruit fly brain is the ideal system in which to do this work. The circuitry is stereotyped between animals. And if we look in a cross-section of the fly eye here, we know the identity of all of the neurons and how they connect in circuits to perform function. For example, here I'm showing you the key neurons that process light increases in green and light decreases in blue. And these neuron populations come in many different sizes. And as I said, we can use tools that are already available for precise genetic manipulation of whole populations. Now, as I mentioned, I want to focus here on the MI1 population. And as I mentioned, there are 750 of these MI1 neurons, and it's hard to observe individual cells if we label the entire population. So I'd like to target a fraction of individual cells that I can easily differentiate and observe in a high throughput. For example, if 50% of MI1 cells express GFP, we can see individual cells, but it might be hard to differentiate between them. But if we only expressed GFP in 20% of these cells, we'd likely to be able to observe large number of individual cells in the same brain. If we only labeled 5% of these cells, we'd see these individual cells, but throughput would be suboptimal as there would be few labeled in each individual brain. Perhaps for a larger fraction of population of cells, 5% might work well. I also wanted the fraction of cells labeled to be precise between animals, so that if I looked at 3 different animals, for example, shown here on the right, I might target different particular MI1 neurons, but I'd always be targeting 20% of them. Okay, so I made this toolkit, and it's a set of transgenes in fruit flies. And here is a schematic of what it looks like. We're calling it SPARC, for Sparse Predictive Activity through Recombinase Competition. And I know that folks in the audience have different levels of familiarity with genetic tools, so let me walk you through this construct and how it works. First, there's a promoter. Here, a UAS promoter, which is recognized by the transcription factor GAL4. Now, in this system, if there's nothing between the promoter and the transgene you're trying to express, then GAL4 will drive expression of that transgene, and here I'm showing you GFP as an example. Now, in flies, GAL4 can be expressed in cell type specific manner, so we can specifically target individual cell populations. But here in SPARC, I put a stop sequence in between the promoter and the transgene. And so this prevents expression of the transgene, and at baseline, SPARC doesn't really do anything. However, I've surrounded this stop sequence by what are called recombinase recognition sequences. And for those of you familiar with Cree, these sequences are analogous to LOXP sequences, except they're recognized by a different recombinase, here Phi-C31. And Phi-C31 selectively and irreversibly recombines one at P sequence with one at B sequence. And so if Phi-C31 is present here, it will selectively remove the intervening sequence shown here, leaving this in the genome, which will enable cell type specific expression of your transgene. And here's another way of looking at this. In SPARC, you get the recombination event here on the top, and it will get expression of your transgene. But as you can see, there's a catch. I added an extra at P sequence in this construct. And what this does is effectively sets up a competition in this construct where Phi-C31 has two options, reaction A shown above or reaction B shown below. Now, if reaction B occurs, the intervening stop sequence stays in the center of the construct, and that prevents expression. Now, this should be deterministic. Only one of these reactions can occur, and once the reaction occurs, the outcome is settled. And that's because Phi-C31 acts irreversibly. Now, all of the different parts of SPARC have been used extensively in flies before, so I next asked whether SPARC works in vivo to give deterministic levels of expression, as you would expect. And I'll use antibody stains from the fruit fly brain to show you how it works. So to orient you, here on the right, you're seeing what a stain from the optic lobe of fly brain looks like, and it's just the tissue here. And we're going to focus on this region of that brain to show you what MI1 cells look like when we use SPARC in the context of that cell population. Now, MI1 cells tile this tissue, as I told you before, and I'm showing you a small section of this on the left, in which I've labeled MI1 with TD tomato through the whole population, and you can see these neurons are interdigitated. Now, when I use SPARC to compete these two at P and at B reactions with each other, I find the pattern of transgene expression shown here on the lower left. And what you can see is you label about 50% of these cells here with GFP. Now, I'll show you more detail later, but these neurons on the lower left are the same neurons that you're seeing in the upper panel in the upper left, and in the lower right, you can see the overlap. Again, this is about 50% of cells labeled, and for time's sake right now, I'll just say that this is consistent across brains. So to recap, if I have two wild type at P recombination sites competing to recombine with one at B site, reaction A happens 50% of the time, reaction B happens 50% of the time in any given cell, and so you get transgene expression in only 50% of cells. Now, we were excited about this, but realized that for some neuron populations, it would be better to label even fewer cells. And so we asked whether we could shift the balance to favor reaction B, so no expression, for sparser labeling. And in brainstorming how to do this, we looked to in vitro assays that demonstrated if you truncate the @P sequence, it's less likely to recombine with @B. So next, I truncated this first @P to see if this increased the likelihood of reaction B so that you had more cells that did not express your transgene. And it worked. When I truncated this first @P from 60 base pairs to 38 base pairs, shown here in the lower left, we label 20% of cells. And when I truncated it further to 34 base pairs, we label only 5% of cells in the population. Now we call these three variants of SPARC the dense, intermediate, and sparse variants. And these levels should cover the range, 50%, 20%, and 5% should cover the range that we need to isolate individual neurons in any neuron population in the fly. But I won't ask you to take my word for it. Here I'm showing you images of SPARC labeling in 4 other cell populations in the fly that span the gamut of cell population size. And we're clearly able to label single neurons in populations from 3,200 cells down to 3 cells with one of these SPARC variants. And next I'll show you how consistent this is within cell type and across cell types. So on the y-axis down here, I'm showing you the percentage of cells labeled by SPARC, and on the x-axis are different cell types for the different SPARC variants. Each circle here represents a count from an individual brain, and here I hope you can appreciate that SPARC dense labels around 50% of cells, SPARC intermediate labels around 20% of cells, and SPARC sparse labels around 5% of cells. Now, of course, here I'm expressing GFP with SPARC, but because the toolkit is completely modular, I can swap GFP for any specific effector that I want and express any other transgene that I want. And I want to show you next how we've used this toolkit for unparalleled experimental precision. And I'll start by explaining how we use SPARC to delete genes from subfractions of these cells. Now, in the interest of time and because it's published, I'll just tell you that my lab mates and I recently developed another genetic toolkit called FlipStop, which conditionally deletes genes in specific cell types and marks mutant genes with TD tomato. This process is dependent on expression of flip recombinase as a transgene. And so therefore, if we express flip recombinase with SPARC in a subfraction of cells, we will mutate a gene in that specific fraction of cells. So here, if I label the whole population on the right with GFP and delete a gene, whatever gene we want, in 50% of those cells, they'll be yellow because they also express the fluorophore TD tomato. Now here you can see an example of this in vivo, in which I deleted a gene from 50% of a cell type called TM3. All cells are labeled with GFP, the whole population, and 50% of them are labeled with TD tomato because I mutated a specific gene here. So just to state it directly, we can now delete any gene out of any cell type from precise fractions of a cell type and compare mutant and wild type genes, mutant and wild type cells right next to each other in the same brain. And in work that I'll be chatting about at my poster, I've used this technique to determine how neurons alter gene expression through single cell RNA-seq. I have been able to look at physiological changes, so how sensory responses change in these neurons using calcium and voltage imaging. And I've also been able to look at structural changes in these neurons as a result of specific gene deletion. Now, this is a very targeted and biased approach, but I also want to show you how one way in which we use SPARC for unbiased discovery of neuronal function. So here, I've used SPARC to express the calcium indicator GCAMP6F in a random 5% or a stochastic 5% of neurons across the whole fly brain. This amounts to several thousand cells, and the sparse expression allows me to image neuronal activity in individually labeled and stochastically labeled processes across the entire brain. Now, I've been working with a grad student in the lab to do this imaging, and he made some movies of neuronal activity in live behaving flies in the absence of any kind of stimuli. I'm going to show you movies from this section of the central brain, and again, the fly is alive and moving under the microscope as we take these videos. So here's the movie, and I'll start it. It covers just about 5 minutes in real time, but we've sped it up. You can follow the timestamp below, and I'll let it go through once. Now, what I hope you can appreciate from this movie is that we can see and track randomly labeled individual processes, and we see activity on different timescales. And because SPARC labels different subsets of neurons in any brain, we can use this tool in an unbiased fashion to scan through single neuron responses in the presence or absence of any stimulus, letting us to associate those neuron responses with a given behavior or response to stimulus. Now, luckily, the fly brain is very highly stereotyped, and we can do post hoc analysis to determine the identity of these specific neurons and discover circuitry that is critical for these different behaviors. Okay, to sum up now. Today, I've explained how the SPARC toolkit works to express or delete any gene in 3 different precise proportions of any cell type. This all happens after the cell has fully differentiated, and therefore we can use this toolkit to look at the role of a given gene in adult neurons. Furthermore, we can express activity indicators in neurons using this method to discover which neurons are active in live behaving flies. And this toolkit has already been adopted by many different fruit fly labs who are tailoring it to their specific needs. Now, I've already started designing a way to translate this toolkit into other genetic model organisms, including mice and zebrafish. And to be clear, these images on the bottom aren't SPARC mice or zebrafish. They're just transgenic mice or zebrafish. But I think SPARC would enable us to do powerful experiments looking at individual neurons in subpopulations of neurons in these vertebrates in particular. And so that's something I'm currently working on. And with that, I'd like to thank my mentor, Tom. He's been incredible in guiding me through this project. My excellent colleagues in the Clandinin lab and my collaborators in the Wilson lab and Mayman lab who are co-authors with me on the recent publication describing this toolkit. And of course, I want to thank the foundation especially for taking a chance on me and giving me the opportunity to explore in this space and grow. Katrina Bryan in particular has done so much on my behalf, and her team even sent me this brainy Beckman onesie that you can see my son Oliver wearing, and it's so good to have him be a part of the family as well. So I'd like to thank you for your attention, and I'd be happy to answer any questions that you might have.